Hey guys, here's the very first podcast for pre-AP biology. We're starting to talk about the nervous system. And so you guys, as we go through this, you can pause, you can start, you can go on. If you need to skip over some things or come back to some things, this allows you to pause and rewind and take notes at your own pace. So, as we go through this, you guys, I want you to remember, life is in the eye of the beholder. The function of the nervous system is to receive, to process, and to respond to information that is received from our sensory neurons. We process it through our brain and spinal cord, and then we have to respond to the internal and external changes through what are called effectors. Effectors are muscles, which voluntarily and involuntarily respond, and glands, which include our endocrine organs that make hormones to respond to internal changes and external changes in our body. The ancient practice of acupuncture actually tries to take into account how our nervous system works. Acupuncture works putting tiny needles into certain spots of the body that will stimulate nerve centers that will then send back information to your brain. The basic unit of our nervous system is the neuron. Neurons are the cells in our body that are going to carry the messages from one part to the other. They're going to carry them from our sensory neurons, our motor neurons, and our interneurons, which are going to connect them to the brain, which will then process it, respond, and send it back. Now, there are 3 different types of neurons, and they all are going to send their messages in the form of electrical impulses. Sensory neurons are ones that are going to send impulses from our sensory organs, our eyes, our ears, the skin for our touch receptors, and our taste buds. Motor neurons are then going to send impulses from our spinal cord to the muscles and glands. They're sending these messages, these impulses, so that the body can respond. And then there are interneurons, which are going to carry impulses from the sensory directly to the motor neurons. This picture shows a neuron in its basic form. Now, neurons have several different parts to them. They have the dendrites. The dendrites, located here, here, and here, are going to be responsible for receiving those messages, these impulses that we call stimuli. And what these dendrites are going to do is they're going to take this message that they receive, and they're going to move the message from the dendrites towards the cell body. Once you get to the cell body is responsible for transmitting that stimuli down the length of the neuron. The length of the neuron, the very long axis center, is known as an axon. This axon is responsible for carrying impulses away from your cell body, carry it to the next neuron. At the very end of the axon are what we call axon terminals. These axon terminals down here are going to be responsible for transmitting that stimuli, that impulse, to another dendrite so that it can continue to send this to the right neurons, to the brain, or to the spinal cord, wherever this message needs to go for it to be processed and then for the body to respond to it. This just sums up what dendrites are at the very top of the neuron, responsible for receiving and moving the stimuli, the impulses towards the cell body, towards the axon, down the axon, all the way to the axon terminals and on to the next neuron. This image here is showing you 2 neurons that are connected together. You have the actual dendrite and the cell body up in this direction over here, the long axon with the axon terminals down here. Once the stimuli reaches the axon terminals, it is then going to be transmitted across what we call a synapse, which we'll talk about in a minute, to the next neuron. These dendrites on the second neuron will then take this message, send it to the cell body, and down the axon of that one. And this will continue so on and so forth until the message reaches the very end, either the brain, the spinal cord, or the neurons that are going to react. These stimuli are going to actually be impulses. These impulses are actually electrical signals that will be transmitted through the neurons from one to the other. The electrical signal is actually created by this concentration of certain ions, certain salts that are set up on one side of the membrane or the other. The actual nerve impulses are done through what we call sodium-potassium pumps. So pumps in the axon in this membrane of these neurons is actively going to transport sodium and potassium back and forth across the membrane of these neurons. The actual sodium-potassium pumps are known for pumping out 3 sodiums and pumping in 2 sodiums. They want no net charge, and the actual potassium leaks back out of the cell membrane of these neurons very, very slowly. Because of this leaking, this results in what we call a slightly negative interior of the neuron. So you have a slightly negative interior and a positively charged exterior of your neuron. The sodium-potassium pumps are set out along the membrane that lines the axon of the neuron. So you're going to have a lot of sodium-potassium chloride outside the cell, which gives the outside this positively charged, it is a weak positive charge. And inside the cell, you have a bunch of potassium that's going to be associated with certain proteins, and this gives the inside of the cell this weakly negative charge. There are some passive membrane proteins that allow for passive diffusion of sodium and potassium back and forth across the neuron. However, when you're ready to send an impulse, it works from one side of the neuron to the other. There are going to be multiple sodium-potassium pumps set up along this axon, and when stimulated, those channels close, allowing the sodium-potassium to go back and forth across freely. And so then, after the impulse goes through, you're pumping out this sodium and letting the potassium come in. So we're causing the membranes to change their charge, and it kind of ripples down the length of the axon. And after it does that, the channels will then open back up, and the cell is going to repolarize so that the outside is more positive and the inside is more negative. Next, I'm going to show you a video of how this impulse works as it walks its way down the neuron.
The cell is less positive than the outside. Two important ions are sodium and potassium. In the cell's polarized state, potassium ions are concentrated inside the cell membrane, and the sodium ions are concentrated mostly outside the membrane. The interior of the cell has a charge of negative 70 millivolts when it is resting. When a stimulus is received by the neuron, the sodium gates open and sodium rushes into the cell. The neuron is now depolarized, meaning the inside of the cell is now more positive than the outside. The voltage of the cell when it is depolarized is around negative 50 millivolts. If the stimulus is strong enough, the graded potential activates the neuron to initiate and transmit a long-distance signal called an action potential. At this point, the voltage is positive. Once an action potential is initiated, it will travel the full distance to the axon terminus. After an impulse is transmitted, the neuron must go back to its resting state. Repolarization resets the cell to its original polarized state. The membrane's permeability changes again, allowing potassium to diffuse out of the cell. The neuron returns to its polarized state in which the inside is negative compared to the outside. At this point, the charges are reset. However, the ions are misplaced and must be switched. The sodium-potassium pump restores the ion concentrations with the help of ATP. 3 sodium ions are pumped out of the cell membrane and 2 potassium ions are pumped in until the ion concentrations are back to normal. The action potential is now on its way to the axon terminus.
As the action potential moves down the axon, this is going to happen over and over again, where we pump sodium in and pump potassium out. This will occur all the way down the axon until we get to the axon terminals. As was shown in the video, action potential is not actually electrical. It's a wave of depolarization and repolarization of the actual axon, the membrane that the neuron shares. Now, as it moves down and you're pumping in and out ions, creating this negative or positive charge, it's going to be either on or off. There is no in between. You can't go halfway down an axon. You either completely go through depolarization or you don't. There is go and stop. And so it's actually really, really slow compared to an electrical current that we know of. The action potential travels about 10 centimeters to 1 meter every second. So electrical currents are much faster than that. Surrounding the axon is what we call a myelin sheath. A myelin sheath is a very fatty tissue layer, and it helps speed up the impulses. Because they are so slow, without this myelin sheath, the impulses wouldn't travel that fast. The myelin sheath then allows these pulses to speed up, and so you don't travel the exact entire space. You skip areas. So you're going through, you travel down one area, and then you skip to the next area, and you skip to the next area. And so it's sending this signal down, and it's almost like skipping a rock. It keeps jumping down the axon in order to get the signal to the very end. What you see in this image is actually a cross-section of a neuron. So we've taken it, we've actually cut the neuron in half, and so we're staring at it from the top. And so what you're seeing right here, this is the myelin sheath, and it's surrounding the neuron. And so this myelin sheath is then actually acting as insulation around this neuron, helping those impulses travel down a lot quicker. Once you travel down the axon and you get to the axon terminal, then these axons can connect to other dendrites, they can connect to receptors, or they can connect to effectors. The receptors are going to be cells in our sensory organs, our sight, our tongue, for our taste buds, our touch receptors, and so they're going to get a signal from the environment. Also, these axons can reach this area where they're going to connect to effectors. So these are going to be our muscles, our glands that produce hormonal responses are going to react to the signal. Once it reaches the axon terminal, it's going to jump the synapse from the one neuron to the next cell. Once you get to the axon terminal, there are going to be little vesicles that are going to hold what we call neurotransmitters. These are chemicals that will travel across the gap, which we call a synapse, between the axon terminal on one neuron and to the either receptor, the effector, or to the dendrite of another neuron. So these vesicles, these are membrane-bound vesicles that are holding the neurotransmitters, which these are going to be hormones and proteins. Neurotransmitters include endorphins, dopamine, epinephrine, serotonin. And so once you get to the end of this axon terminal, the axon potential is run all the way down. It gets there, and these neurotransmitters get released into the gap between itself and the next neuron. And so they get released into the synapse, they travel across the synapse, and on the other side, these neurotransmitters are going to open the sodium channels and start the depolarization and allow that action potential to move on to the next neuron and travel on down. This continues on and on until you reach either the effector, the muscle, or the gland, or some other type of sensory neuron. This is just another image showing this axon terminal and with the synapse. So what we are is we are right here, we're between one neuron and the next, and so what's happening is these neurotransmitters are being released once the action potential reaches the neuron, released into the synapse, this gap down here, and they are going to bind with a receptor on the other side that allows for them to open up channels, specifically the sodium-potassium channels, and allow for that action potential to continue. Dopamine is one of the master neurotransmitter molecules in our nervous system. And so dopamine is actually thought to be a master molecule when it comes to addiction. After release, dopamine gets quickly absorbed or it gets broken down by an enzyme called MAO. That stands for monoamine oxidase. And so when dopamine is released, it's usually released after eating dessert, having sex, other types of pleasurable activities. Dopamine gets released out, and so it's either quickly absorbed back into the neuron that it's released by, or it gets broken down. So for a while you get this euphoria feeling after you've had some type of pleasurable activity. However, because it gets broken down or reabsorbed very quickly, that pleasure feeling, that euphoria, only lasts for a short amount of time. This is specifically important because drugs that we take that will give us those euphoria feelings are going to mimic dopamine's effects. So what happens here is cocaine. Cocaine is one of those drugs that what happens is it blocks the reabsorption of the dopamine. So as the dopamine gets released, it gets stuck in that synapse area, and then cocaine comes in and blocks them from being able to get absorbed back into the membrane, into the neuron it came from. Because it cannot be reabsorbed, then the dopamine causes this prolonged sense of euphoria. Once cocaine has been degraded, then you lose that euphoria. For amphetamines, specifically we're talking about things like meth and ecstasy, amphetamines are going to stimulate excessive release of dopamine. So you're going to release too much, more than enough. And so what happens when you release a whole lot extra of the dopamine? It overwhelms these neurons, and the neurons are having trouble reabsorbing it quickly, and there's not enough of the MAOs to actually break it back down. So again, we get this net result that we have this elevated level of dopamine, we get this high, pleasurable euphoria feeling, and once then the meth or the ecstasy, the amphetamines are then broken down, all the dopamine is reabsorbed or broken down, we lose that feeling. And then nicotine works the same way. It also works that it stimulates excessive release of this dopamine, but it also does the same thing cocaine does, in that it blocks the ability of it to be reabsorbed or broken down. Specifically, nicotine is looking at blocking the ability of the MAOs. So they're going to block these enzymes that break down dopamine, and so this leaves the neuron ability to reabsorb it, and that has to catch up. So you get a very short-term high, but the neuron's got to reabsorb all that excess that it released. And so nicotine blocks the MAOs to break it down. Cocaine blocks the reabsorption process. So they're a little bit different. Now, as you guys could probably guess, as drugs enhance the amount of dopamine that is in our synapses, giving us euphoria feelings, increased dopamine, increased feelings of pleasure. And so once our cells get used to these levels of dopamine, then we start considering them normal. So after a while, taking the same amount of drug, our brain begins to realize that we're releasing excess or we're not reabsorbing it. And because of this, our brain is going to react. And what it does, it reacts by reducing the number of dopamine receptors on the neurons. So we're doing this negative feedback where we're trying to keep things in balance. Because of the drugs, we're going to decrease what we can respond to. Because we decrease the number of receptors that we can respond to the drugs, then if you're addicted, you have to take more of the drug in order to continue to get that euphoria feeling. As you increase that number, again, your body's going to react and decrease the receptors again. And so this process continues with the number of receptors decreasing to the point that eventually the drug addict has to take high levels of the drug just to feel okay. Not to feel bad, but just to feel okay, they have to take the drug because you've reduced the receptors that our body can normally respond to. Now back to the nervous system. In the nervous system, there are two major divisions. We take the brain and spinal cord and we call that our central nervous system. And then we also have the peripheral nervous system, which is abbreviated PNS. And those are going to be all the other nerves, the receptors, the sensory neurons. They're going to come away from the brain and from the spinal cord. So you're going to reach out, and they're going to be our motor and sensory neurons. In the central nervous system, the spinal cord is one of the main pathways that links all our receptor cells, the senses, the effectors, to our brain. So it is the direct link. It's our communication link. And so this communication link, it actually regulates the reflexes. And so our brain doesn't have to process reflex motion. That's why it's so quick. If you hit yourself on the kneecap, you jerk and kick, because the response is going to the spinal cord, back to the muscle, in order for you to get that reflex response. So the spinal cord is trying to help out the brain and trying to ease up on everything it has to process. The brain is going to do a bunch of processing. Our brain actually contains approximately 35 billion neurons. Now these neurons are going to be very protected. They're protected by layers and layers of this tissue, which this tissue is called meninges. So if you guys think back to our infectious disease lectures where some people talked about meningitis, meningitis was the swelling of the layers around the brain. These meninges are there to protect the brain. And so there are three meninges, three layers of tissue that wrap around our brain. The pia mater, the arachnoid, and the dura mater. The pia mater is directly bound to the brain surface. There's lots of blood vessels that go to the pia mater, and so it allows for that blood-brain barrier to be overcome. The arachnoid layer is outside of it, and it's another layer, but this layer is filled with fluid. So that way when you fall and you hit your head, there's some shock absorbance there. And then the last one is the dura mater. It is the one on the outside of the brain. It is a very thick layer, and it's also fluid filled. And so again, we're just packing it so that way we have this protection of the most essential organ, which does the processing for everything but our reflexes. Here's a picture of the brain. And so here's your brain. And surrounding the brain, we are going to have the meninges. So the first meninges is going to be this pia mater, which is this very thin layer running right across here. And so it's just the layer, it's protection, it's like shrink wrap around the brain. In between the pia mater on the outside, then you have this fluid-filled area right here, which this is your arachnoid region. Again, this is for that shock absorbance, so if you hit your head somewhere or something like that, we're not going to damage the brain too much. Now, if you hit your head, you are going to endure some type of brain damage, just not a lot. And lastly, we have the dura mater, which is this blue layer thinly on the outside, and there's going to be fluid between it and the arachnoid. And so that, again, is another just layer there to protect it and allow the brain to hopefully allow itself to move around and respond in situations where it's stressed. The brain is going to be responsible for a lot of different functions. Our cerebral cortex is actually part of our higher level thinking. This is what gives us the ability to think critically, do problem solving. It's also the thing that controls our movement, our motor skills. It allows for our sensory neurons to send impulses here for us to be able to know what something that tastes salty or sweet is. And it allows us to speak. Also in the brain, the circadian rhythm is controlled, and so sleep is the lowest possible activity level of the cerebral cortex. So even while we're asleep, our brain is still functioning at a minimum level. And of course, we need the sleep in order to help repair and overcome some of the stresses that we had in our daily activities. And then the last thing is the memory. Memory is short and long term, and they are stored differently. Short-term memory and long-term memory are stored in different locations. And if you guys think back to 50 First Dates, where she had lost her short-term memory, she could lose her short-term memory without her long-term because of where they're located. You could have damage to one part and still remember all the long-term memories, or you could have damage to the long-term memories and still be able to absorb and keep short-term things. This is the real image of the brain. So as you guys see, there's going to be several layers, and down here is where the brainstem is going to connect to the spinal cord and run down the rest of the body. Here is a cartoon image of the brain showing where our frontal lobe is, and it's also showing each area that's involved in consciousness and speech and motor movement. And also we can see that there's a specific section right here that is what we call the somatosensory cortex. Those are where we're going to feel those senses, and it's where our body's going to process them and then return a type of stimuli back to those cells. Now what you guys see here, this is what we call a somatosensory map. And so somatosensory maps are point-to-point correspondence of an area of the body to a specific location in our central nervous system. So we're actually looking at that somatosensory cortex, that very thin layer. We've cut it out and we've turned it on its side. And so what we are showing here is that we're showing the area of the body that corresponds to the point on the cortex. So we're representing them as what we call a sensory homunculus. Homunculus is you're taking a tiny person and you're extending specific body parts to the representative locations where they are processed in the brain. So here in this image, you see things like your hand, your senses for touch and all that from your hand are going to be processed in this area of the brain. The face gets processed down here, and so your leg gets processed up here. So each section has its own area where it's being processed. Now, things that are more finely controlled, like your digits, you see that those are extended out over a really lengthy period in your cortex. And so that's because they're finely controlled. We have to do a lot of fine adjustments. The things that aren't as finely controlled, they're coarsely controlled, like our stomach, our trunk region, things like that. They're going to have smaller regions because it doesn't take as much to process that information. The peripheral nervous system, or PNS, is going to be the actual division that involves our sensory neurons. And so they're the motor neurons, the effectors, those muscles and glands that we are responding to and impulses are coming from. Our peripheral nervous system is divided into two parts. You have the somatic nervous system, which is us consciously thinking and doing something like writing or speaking. And then we have our autonomic nervous system. This is automatic, involuntary movements that we don't control, like breathing, our heart beating. All of that's going to be controlled under the autonomic nervous system. If we look even further at the autonomic nervous system, there are two parts at it. Two parts of the autonomic nervous system, they have opposite functions. One is going to speed up, and so the ones that speed up, these are called our sympathetic nervous system. And then the other part is our parasympathetic nervous system, generally involved in maintenance of the body. So we have an accelerator and a brake involved in our autonomic nervous system. Our body's naturally going to respond to things, and then it's going to either speed up or slow down in response to what signal we're getting. What this image is showing several different divisions of the parasympathetic and sympathetic nervous system. Parasympathetic, again, remember that is where our body is going to maintain, it's going to be the brake part of it. So our bladder, the release of bile after digestion, are constricting our bronchi, which is going to constrict our lungs, how much we can breathe, or slow our heartbeat. That's all the parasympathetic. That's normal daily activities that we're going to be involved in. Now, sympathetic nervous system, this is going to speed things up. So when we get excited, that flight or fight sense that we remember. So our pupils dilate. Our heartbeat accelerates. We're going to increase the rate that we're breathing. We're going to take glycogen, which is the storage of glucose. So we're storing energy, and we're going to start converting it to glucose for us to burn that. And then it's going to do the secretion of the adrenaline and noradrenaline. So again, we're going to start having that fight or flight sense with our sympathetic nervous system. It also inhibits the bladder contraction, so that way we don't have to go to the bathroom when we're in the phase where we need to respond and run away from a situation. Our sensory organs, these are going to be the sense organs like the sight, the taste, the touch. And each of these have their own receptors that take that stimulus that we receive, if we eat something, if we hear something, and it sends it to the brain. So each sense has its own sensory organ. You guys can probably pretty much guess what its sensory organ is. You think we're almost at the end, but we're not quite there. So what we're going to do now is we're going to take on and we're going to look at each of those senses in deeper. And so you guys, what I really want you to take in is you've been using these senses this entire year. And because you've been using these sensors, these are ghosts of lectures past. You've been using your sight. You've been using your hearing. You've been using the touch during our past lectures. So the first thing we're going to talk about is vision. And so vision happens when light is entering our eye. It enters the cornea of our eye. And from there, it's going to pass through the pupil in the center of the iris, passes all the way through, and gets back to the retina behind the eye. Once it hits the retina, back in these areas we have receptors. They're photoreceptors, our rods and our cones. And these rods and cones are arranged in layers so that it can detect the light, and then it's going to send those impulses through our optic nerve all the way to the brain for it to be processed, and then it's going to come back, and we're going to respond in some manner to what we just saw. This image breaks down the actual eye. Here you have the pupil, which is the area in here. The actual iris is this area outside. It's your colored part. Pupil is the dark center. The cornea is a layer that surrounds your eye. You have an eyelid that covers it. We're trying to keep things out of the mucus in the eye. And the light's going to travel through the pupil all the way back, and it's going to hit this back retina area. This is where our rods and cones are going to be. Once it hits here, those receptors are going to send the signal through the optic nerve to the brain. Our hearing. our hearing is connected to balance. And so we have canals in our ears, and these are semicircular, and they act as what is like a gyroscope in our inner ear. Sounds get funneled by the outer ear through a canal to our eardrum. This eardrum, like a drum, vibrates when it gets hit with sound. And when it vibrates, there are 3 tiny bones in our middle ear. And these 3 tiny bones hit the membrane between the inner ear and the middle ear. So we're translating this actual vibration sensation along the inner ear. And so when it hits, there's fluid in what we call our cochlea. Cochlea are these fluid-filled sacs that have the receptors. And so when this vibration passes through the middle ear to the inner ear, and it hits the cochlea, these vibrate and stimulate these tiny hairs that are inside our ear. There's also a reason you don't want to use Q-tips to clean your ears out, because if you stick Q-tips into that canal, you're going to pack back the earwax, and then that's going to block the sounds being able to vibrate that eardrum. So then you're going to lose senses of your hearing because you've packed another layer between where the sound's vibrating through. And so these sounds won't be as crisp as they would be if you cleared your ears with earwax candles or through other methods. Smell. Now smell, we have chemoreceptors, so these are chemical receptors in our nasal passages, in our nose. And so they're going to detect chemicals that are in the air surrounding us. Now, problem with smell and why it's going to be so brief is no one really knows how individual smells are distinguished from one another. We don't know if there are different receptors for each type of smell, or if the receptors respond differently to the different chemicals that get bound onto our receptors. Now we have taste. Taste is one of my favorite. I like food. I enjoy food. Chemoreceptors, again, we are looking at the chemicals in our food that are going to stimulate our taste buds. And so our tongue actually detects sweet, salty, bitter, sour, and then there's also another one that the tongue is going to detect, and it's called umami. And so these allow us to detect the different things. Umami is like a meaty sensation, and so this allows us to enjoy our food. However, taste works with the chemoreceptors that are involved in smell. So if you can't smell, you can't taste. And so you need both of them working together in order for your taste buds to respond and send that signal to your brain. Alright guys, we're at the last sense, touch. And now touch, the organ for touch, that's our skin. All our skin is wired for touch sensory. It's the largest of all our sense organs. It wraps around us. And there are several different types of receptors to respond to different touches we get. You have types of warm and cold, and there's actually more warm sensors than cold in the skin. There's also receptors specifically for touch. Pressure, touch, all those kinds of things have their own receptors. And then there's one type of receptor for pain. And so this is going to allow that prickling, burning, aching sensation that we get when something pokes us or we scrape ourselves. And this image shows your skin. Your skin's composed of the epidermal layer, which is going to be a very thin layer of epidermal cells, and you have the dermal layer. And so these receptors, you're going to have heat receptors in here, you're going to have light touch receptors, and then you're also going to have deeper receptors for the for harder touches. Pain is near the surface. We have some cold receptors as well. And so all of these are in that dermal and epidermal layer, allowing for you to feel all these different sensations on the skin. Alright guys, we've reached the end. I've put in some review questions since we didn't actually get to talk in class. I would appreciate you guys working through them, write them down. Make sure you add a summary at the end of these notes. And when you guys finish, please leave me any information that you need. Did I talk too fast? Do I need to slow it down? Did this work really well? Or did you have trouble accessing this actual PowerPoint? Let me know. Hope to see you guys in class soon.